Next panel comes forward and gets seated. A couple of more logistical points for you, and I thank everyone that have come into the room recently. This is a nice audience. We're going to be having a plenary discussion next, and afterwards, at approximately 10:35 or so, we're going to have our first break. And immediately outside in the Civic Auditorium, there'll be coffee in Danish so everyone can get nourished and be able to go the rest of the morning. And then at 1 o'clock, there will be something we call Conversations with Experts Luncheon. And many of you, upon registration, registered for your box lunch. This event is sponsored by EMD Millipore. And it's very, if you haven't participated before, it's one of, generally we get tremendous comments because this allows the attendees to join in on some 40 to 45 tabletop conversations. You grab a deluxe box lunch, you will get a sheet showing an expert, a topic, science and industry, patient focus, science society, industry, regulatory, and you get an opportunity to sit with an expert and others interested in this field and have a guided conversation and network with people. And it's an opportunity to meet someone that is a leader in the FDA or a top researcher or a patient advocate or a government leader. It is first come, first served, and you have to think on your feet and find the right table. If you find a speaker sitting all alone, it's up to you not to make that speaker feel lonely and sit at the table. And if you are shut out of the topic that you want, you can learn something new. And finally, if every seat is filled and there's not a topic you're interested in, there's plenty of seating and you meet with your fellow attendees. Even though some of you may not have your badges yet, you have to be a little more extroverted. You have to introduce yourself. This is fine. So our next panel is coming out and is going to sit up right now, wherever you are. Please take your seat. In a minute, I'll introduce the introducer. After the speakers. All right, we're going to take it one by one. They're not going to sit. Generally, I'm the least informed person in the room. We're going to handle it a little differently for this panel. So I want to introduce first are one of the co-chairs of the World Stem Cell Summit that will provide the initial introduction of this panel, and that is Dr. Clive Svensson, the director of the Regenerative Medicine Institute of Cedars-Sinai Hospital. Clive Svensson, as you know, was a past co-chair of the World Stem Cell Summit in 2008 when he was at the University of Wisconsin in Madison. He re-upped again. He is a distinguished member of the Science Advisory Board of the Genetics Policy Institute. So I'd like to introduce Dr. Clive Svensson.
Thanks very much, Bernie. Very nice to be doing this again. I can't believe I'm actually co-organizing twice in a row for all my friends from Wisconsin. Look, I did it again, second time. I'm very happy to introduce this panel, a very important panel. It's the International Society of Stem Cell Research that supported this panel. The title is How Stem Cells Are Transforming Medicine. The ISSCR was set up 10 years ago, actually, exactly. It's a non-profit organization. It's to foster exchange of information on stem cell research. It's had a very rapid expansion. There's more than 3,500 members of the ISSCR, and it's a phenomenal exponential increase. It reflects the interest in stem cells, not only in America but around the world. It attracts over 3,500 folks to its annual meeting. I'll give one plug for that. It's in Yokohama, Japan this year. It's the 10th anniversary of this meeting, so it's a big event for them, so it's very exciting. And finally, the website is fantastic. I recommend patients with ALS who come up to me a lot and say, where can we get treatments outside of America? I always direct them to this website, it's ISSCR.org, because they have a fantastic part there for the public section, and they've got a whole section on stem cell treatments. And I think It's one of the best places to go look if you want to have information on these stem cell clinics outside of America. So I'm very happy to introduce them, and I'm very happy that they supported the World Stem Cell Summit. So I'll now pass over to their science director, Heather Rooke, who's going to introduce the panel. So thanks very much and enjoy the meeting. And Heather?
Thanks. So this morning we're going to hear from 3 of our members about how stem cells have impacted their research and discovery and the progress that they've seen around them. So we're going to hear their perspectives. I will first introduce each of the speakers, ask them to talk, and then after the final speaker we'll come out, have a panel discussion, and we'll open the floor to some questions. I would first like to introduce Anne Tsukamoto, PhD. She's the Executive Vice President for Research and Development at StemCells, Inc. She is responsible for StemCell Inc.'s scientific and clinical development programs. This company is engaged in the research, development, and commercialization of stem cell therapeutics for central nervous system and liver disorders, and offers tools for use in stem cell-based research and discovery. They have a number of stem cell therapy clinical trials that are underway based on this product. But prior to joining StemCells Inc., Dr. Tsukamoto worked with Systemix, an early stem cell company, where she played a leading role in the launch of the clinical research program for a blood stem cell product, one of the prototypic stem cells in the clinic. So we look forward to hearing more from Anne about how stem cells have been moved into the clinic and her perspective on the path ahead.
Thank you, Heather. And I'd like to thank the organizers and the ISSCR for the opportunity to speak. I'm one of the old folks in the field. I've been involved in stem cell research for the past 22 years. And so I thought I would talk about how stem cells can transform medicine because I think we still have quite a ways to go in this field, as you've heard from Alan and some of the distinguished speakers at the beginning. So I thought I'd give you a bit of history, since I've been involved in this field for such a long time, and just talk very, very briefly about where stem cell therapy could be today if some of the events that transpired didn't happen. And then I'm going to talk about the clinical trials with our human neural stem cells, what's completed and what the lessons that we've learned from that first trial. And then what's in store for the future, what's in progress, and what we are learning today. So just as a brief overview, if you look at the translational pathway, as you've heard from Alan's talk, there are a lot of things that have to line up essentially for this moon launch. And in order to bring cell-based medicines to clinical application for the whole spectrum of diseases, you need to define which cell you're going to work with. You have to identify the right cell. Does it have the right bioactivity that you're looking for? And can you overcome the technical challenges? So if you start with the stem cell, which, as you know, has very unique properties to self-renew and make all of the organs of the mature cell lifelong, this cell may have the right properties that you're looking for. So to a large extent, the bioactivity that you want may be disease dependent. And so if you want lifelong benefit or long-term benefit, you may need both survival and functions of the cells long-term, and not just a transient production of trophic factors. So you really have to define your disease target and what you need. And I'll talk a little bit about the specifics of that when I talk about our spinal cord injury data. And then in terms of technical challenges, you have to identify and purify the right cell, and you have to be able to manufacture these cells in sufficient quantities for clinical applications under GMP conditions or good manufacturing production conditions. And then you need to have the right assays for the cells. So before you inject the product, you want to know that what you're injecting is really what you were working with in the research lab. So you have to characterize the cells to the extent that you know what you're putting in. And only then, after you achieve all of these things, do I think we're going to get to the point where we are going to have long-lasting benefits, and that stem cells and cell-based medicines can be a transformative therapy in diseases such as diabetes, spinal cord injury, Alzheimer's disease, and macular degeneration, just to name a few. So my experience started with the hematopoietic stem cells, as you heard from Heather, at Systemics. And this was a company that was founded by Mike McCune and Irv Weissman from their work that was done at Stanford University. And Systemics was formed based on the discovery in Irv Weissman's lab on the purification and identification of the mouse hematopoietic stem cells, and also the creation of the SCID-HU mouse model. So Systemics' mission was to purify and identify the human counterpart, the human hematopoietic stem cells, and take that to clinical application, and as well as to use that as a tool to discover HIV therapies, or possibly HIV gene therapy, protecting the hematopoietic stem cell and their progeny. So in 1989, the company was formed, and in 1992, just 3 years later, we identified and purified the human hematopoietic stem cell. And this, we did a lot of preclinical studies to demonstrate that this was the right cell based in the mouse model, and started clinical trials in 1996, 4 years later. So from start of the company to actually starting the clinical trial was about 7 years. And then because of a number of events, a company was purchased by another entity, and they decided that even though the clinical data looked promising, that it was an autologous patient-specific product, and that the company would close because it wasn't going to be easy to translate this to a large application throughout the community. But just recently this year, 14 years later, some of the investigators involved in that study at Stanford published a paper on the long-term follow-up of the breast cancer patients that were enrolled in this trial. And on the next slide, you're going to see some of the data. And what it shows is that using cancer-free purified hematopoietic stem cells, you can have improved survival of these patients. And these were stage 4 metastatic breast cancer patients who had failed all other therapies. So they had metastasis to the bone and to other organs. And so they were enrolled in this trial. And what you're seeing here on this graph is that, the patients who received, the patients in the blue line who received the purified graft and high-dose chemotherapy, compared to the black line, which are patients who received the same high-dose chemotherapy but non-purified grafts, there's a threefold difference in the survival rate after 14 years. So what have we learned from this experience? We learned that these purified hematopoietic stem cells could survive long-term in the patients, and that they reconstituted the blood system for at least 12 years. And so this cell population was the right cell, and it self-renewed and maintained all of the blood cells. Moreover, the grafts that were depleted of the cancer cells can slow or stop the disease progression, and I think this is a very important point. But during the intervening 14 years, I think if we had the right partner, the therapy could have been developed to treating some of the cancer therapies that Alan had on his slide, or it could have improved or cured the treatments for some of the genetic disease that he also had. We also know that hematopoietic stem cells are the key to transplantation tolerance for allografts and for organs. And so if this research had continued, could we now be at a point where we would have transplantation tolerance to organ or stem cell therapies? And that question we won't have the answer to. So now I'm going to switch gears and talk a little bit about our human neural stem cell work that we've been doing at StemCells Inc. And this is a highly purified cell population. It's non-embryonic and unmodified adult stem cells. It's expandable to commercial scales, so we can make large banks of cells to treat the patients. And these cells are restricted to the CNS lineage, so that when we look at the cells once they've engrafted in animal models, they make all of the cell types of the central nervous system, neurons, astrocytes, and myelin-producing oligodendrocytes. And to date, and we've transplanted many, many animals and some patients, they have not formed tumors or any abnormal growth, so we believe they are non-tumorigenic. In vivo, I'll show you on the next slide, the cells engraft, migrate, and have site-specific maturation. And these cells are able to both neuroprotect and in cases replace lost neuronal cells. So the potential application, there is potential application to a broad spectrum of CNS disorders. And what I'm going to do for the rest of my talk is just very briefly touch on the preclinical data that we've obtained to support the clinical trials, or where the preclinical data is currently to support future trials. So if you look at the timelines for the human neural stem cell, the StemCells Inc. was founded in 1998. We identified the cell in the year 2000, the paper by Uchida et al. And in 2006, we transplanted our first patient in NCL, or Batten's disease. But unlike systemics for the last 5 years, we've been continuing to plow ahead very slowly, albeit, and we've made, I believe, significant progress in translating these cells to clinical applications. And I'll just make a note that Dr. Hune, our head of our CNS program, will be talking at 1 p.m. on Wednesday in a session entitled Promising Science from Industry. And Martin McGlynn, our CEO, will be talking at 2 p.m. on Wednesday on key clinical trial updates. So a key lesson that we learned from the hematopoietic stem cell was what the cells do in an in vivo animal model. And what we've been able to show with these purified, expanded, and banked human neural stem cells, that once transplanted into the brain or spinal cord, the cells survive long-term, they proliferate slowly, and they migrate throughout the tissue. And so what you're looking at in this slide is a cross-section of brain, and all of the brown staining you're seeing are the human neural stem cells that have been grafted, and they migrate throughout the brain, and they land in specific sites where they differentiate in this site-specific manner. And here you're seeing the cells which have become granular neurons in the olfactory bulb. And on this slide, you're seeing that the cells become neurons, astrocytes, and oligodendrocytes. So how have we applied these cells to the clinic? So the first one I'll talk about is our strategy to enter clinical testing. Now remember, nobody has ever put purified and expanded and banked neural stem cells into any CNS tissue. And so we had to start with very fatal diseases. So we started our clinical trial with a disease called Batten's disease. It's a rare lysosomal storage disease. And we moved into another genetic disease called Pelizaeus-Merzbacher, which is a myelin disease. And we just initiated our trial in spinal cord injury and transplanted our first patient, which you'll hear about. And upcoming for the future are age-related macular degeneration, Alzheimer's disease, and potentially stroke. So our first-in-man studies in Batten's disease, as I mentioned, it's a rare, fatal lysosomal storage disease which lacks a specific lysosomal enzyme. So the cellular waste material builds up in the brain, the neurons, and it causes their death. And the cells, the human neural stem cells that we've purified, produce this enzyme naturally, since it's a housekeeping gene. And the production of this enzyme can cross-correct the deficiency in animals. So we initiated a phase 1 open-label study at Oregon Health Science with Bob Steiner and Nate Selden. And these enrolled 6 subjects with either infantile or late infantile disease. And what you're looking at in the chart is a graph of the chronological age versus the developmental age. So the first subject who you see the MRI on the top was the first subject transplanted, and the subject was chronologically 7 years of age with a developmental capacity of less than a 1-year-old. And you can see by the MRI image that the brain is very atrophied. The enlarged ventricles, which are the 2 butterfly-shaped shapes in the center, are the ventricles which are normally much, much smaller. The last patient enrolled in the study was much less progressed in the disease course, except still very severe. The subject was 9 years of chronological age with a 5-year developmental age. And these are the MRIs pre-transplant. And post-transplant, you can see that the disease progression continued, not so much in the case of the last subject, but definitely in the case of the first subject. And so the lessons that we've learned from this first trial, which completed in 2009, is that direct transplants of large doses of human neural stem cells, up to 1 billion cells, into the human brain was feasible and well tolerated. 12 months of immunosuppression in these subjects was well tolerated. No tumors were observed, and there were no other abnormal growths. The cells migrated, engrafted, survived, and migrated deep into the brain. We have long-term survival data of the cells out 2 years in one of the subjects who died, and we did an autopsy. And moreover, we've shown that 1.5 years after immunosuppression withdrawal, we still detect the human cells. And so I think that is very good to know, that what we had predicted from the animal studies, that the cells engraft and migrate, is happening in the human subjects. But we also learned that trial in rare pediatric disease is problematic. And one of the issues is that we need earlier diagnosis. We started a phase 1B subject in children who were less impacted, but we didn't enroll any subjects because most of them were too far progressed in their disease course. So earlier diagnosis is really key to helping move forward in this field. So the next clinical target and area we embarked upon were the human neural stem cells in myelin disorders. And as you see, the cartoon on the top shows a picture of a nerve axon in the myelinated axons. And what you're seeing is that axon is wrapped by a myelin sheath, or the insulation. And that insulation is provided by the oligodendrocyte. And in the bottom 2 panels to the one on your left, what you're seeing in green is the human myelin that was produced, or the myelin sheath that was produced by the transplanted human neural stem cells into a mouse model which doesn't produce myelin. And in the center is in the red in the center is the mouse axon. And that was shown that the myelin was a normal myelin wrap by the electron micrograph in the black and white figure adjacent. And what's exciting about this disease area is that there may be a surrogate marker for bioactivity. We are hoping that evidence for myelin production can be imaged by MRI because it can be done clinically. And that we entered a clinical trial in Pelizaeus-Merzbacher, the hereditary leukodystrophy, and we hope that this might serve as proof of principle for other myelin disorders. So here is a little bit about the disease. It's a mutation of the major CNS myelin protein, which is the PLP protein. And the clinical diagnosis, it's an X-linked disease, and you see diffuse white matter abnormalities by MRI. And the disease is severe and progressive in neurological deterioration. So what you're seeing here is an MRI image of a normal 2-year-old brain, and in the your right panel you're seeing the image of a PMD subject at the same age. And what you see pointing with the red arrow are the black area is the myelin in a normal brain, and you can see that that is absent in a PMD patient. So our phase 1 study at UCSF under Dr. David Roach is a phase 1 study enrolling 4 subjects. We have completed the dosing of all 4 subjects. It's a single dose of 300 million cells. The study is expected to complete in February of 2012. We're currently performing interim analysis. And the goal of this study was to demonstrate safety and donor myelin production by MRI. So stay tuned for the results of this trial. Another area where myelin restoration would be helpful is in spinal cord injury. And we've shown with our collaborators, Eileen Anderson and Brian Cummings at UC Irvine, that the human neural stem cells can restore motor function long-term in both subacute and chronic spinal cord injured subjects. And the mechanism of action is through remyelination and possibly neuronal bridging. And what I'll show you in the next slide is that the presence of human cells is required to maintain that restored motor function. If you lose the human cells, you lose the motor function that was restored. So we've initiated a phase 1-2 trial in Switzerland that will enroll subjects that are the most severe, Asia A's and B's, and then subjects who are less severely affected, Asia C's. And we transplanted our first patient in this trial last month. So the data supporting this clinical trial is shown in this slide. And what you're seeing is a graph comparing animals who received a control injection of either cells or media and animals who received transplants of human neurostem cells. And all of the points marked with an asterisk show a statistically significant difference in the restoration of motor function. The investigators went on to ask another question, which is, are the human cells required for this to maintain this restored function? So what they did is they took all the animals in this study that had restored motor function, they split them into two groups. They treated one group with saline alone, which is not expected to do anything. And as you see, those animals that received saline maintained their human stem cell graft, as shown in the top histology slide, as well as maintained their motor function. And in contrast, when animals were given diphtheria toxin, which is a powerful toxin that will selectively ablate human cells in this mouse model, you can see that not only do you lose the human cells in the graft shown at the bottom, but you also lose the motor function that was restored. So this is an example where the presence of the human cell is going to be required for maintaining that restored motor function. So a transient introduction of cells that produce factors will probably not keep the function restored. So I'd like to now move to what's in store for the future. So we have announced that our next clinical target is age-related macular degeneration, and we're preparing to file our IND. And what we've been able to show in collaboration with Ray Lund and his group at OHSU was that the human cells transplanted into the subretinal space of a rat model called the Royal College of Surgeons rat are able to migrate throughout the subretinal space, protect the photoreceptors, and slow the progression or prevent the loss of vision in these animals. And what you're looking at in the graph is essentially a functional test, which is equivalent to the eye chart that we do when we go to the eye doctor. And so we perform this on the rats. And what you see in the blue and the red line are the animals that received either media alone or were unoperated and received no treatment. And in those animals, you see a progressive decline of their visual acuity. And in contrast to the animals who have received human neural stem cell graft at postnatal day 21, which is when the photoreceptors start to decline. And as you see, that we're able to preserve the vision that would normally be lost up to 240 days out post-transplant, which is the longest time point we've tested. So this slide shows you the eye cup, and it's very hard to see, and I'll go into higher magnifications. But what you're looking at in that purple line are the human cells that are having grafted and migrated in the subretinal space between the photoreceptor layer and the pigmented epithelial layer. And what we believe is happening is that these human cells are phagocytosing the shed outer segments and clearing the debris zone that normally happens in this model, and they're protecting the photoreceptors through that one, as that is one of the mechanisms by which they may be protecting the photoreceptors. In the next slide, what you're seeing are published studies on the RCS, the photoreceptor degeneration in the RCS rat. And what I'm pointing to is the red circle. And what you see is that by postnatal day 90, these rats essentially have one cell layer of photoreceptors compared to the 8 to 10 when they're born and at postnatal day 21. And by P240, you see that the retina is very degenerated and disorganized in these rats. So when you compare what's happening in the animals that receive the human neural stem cell transplants, what you're looking at on your left is the animals that were at P90 post-transplant. And what you see bracketed in between the red lines are the photoreceptors that were protected in this animal model, and they did not degenerate, coinciding with the preservation of visual acuity I showed you in the graph. And in postnatal day 240, you again see the human neural stem cells have protected the photoreceptors. What we know from a more quantitative analysis that the cells that are being preserved in this area are the cone photoreceptors, which are the key cells that are lost in humans in age-related macular degeneration. So we're furiously working on our studies to complete to file the IND, hopefully before the end of this year. And so a disease target that we have recently received CIRM disease team planning award for is in Alzheimer's disease. And the data we have to date suggesting that the human neural stem cells might have an impact in this disease comes from two sources. One is some in-house studies we did in collaboration with George Carlson at the McLaughlin Research Institute, showing that the human neural stem cells survive in a plaque-riddled environment. They're not destroyed by the plaqued environment. And the second comes from Frank LaFurla and Matthew Burton-Jones at UC Irvine, where they published that the mouse neural stem cells transplanted into their mouse model of Alzheimer's disease are able to improve cognitive function. So we are now collaborating with Frank LaFurla and Matthew on our CIRM disease team planning award to evaluate the human neural stem cells in their Alzheimer's disease mouse models. So what I don't have time to go through today is all of the scientific data supporting our march to the clinic. I discussed with you the 5 areas that we have pursued or completed or are currently pursuing in clinical trials and our future trials. But every time we start one of these trials, it is like launching to the moon because everything has to line up just exactly. We have to have the cells, the preclinical data, and then we have to have the clinical sites, the patient advocates, the funding for the trials. So many things have to line up to make these happen. And I just want to conclude that in summary, the stem cell therapies can transform medicine, but we need, as Alan mentioned, working together, the scientists, the clinicians, the regulators, the advocates, the funding agencies, and the investors. It isn't a short runway that we have to bring this to fruition. It does take a long time. There are many things that have to line up, and we all have to work together to see this happen. We have to make sure that medical tourism with unproven therapies doesn't continue and eclipse the good scientific work done by a lot of people here in this audience. There are many articles every day. The demand for stem cells is growing. Many turn to medical tourism. Very famous people are going to other parts of the world for their stem cell therapies. And if you think a Hail Mary pass in football to get the cell into the end zone is important, some of our famous football players are going to other entities for their Hail Mary pass for a stem cell treatment. So I believe that rigorous scientific data is key. And I'll leave you with this final thought, is that for us wine aficionados, there is a saying, in vino veritas, in wine there is truth. And I'll leave you with our one of the models that we live by at the company, in vivo veritas, truth within the living. Thank you.
Thank you, Anne. And I think that this is an issue that you'll see touched on in a number of places during the meeting. It's certainly something that we are very concerned about. I would now like to introduce you to Arnold I would now like to introduce Arnold Kriegstein, who's the director of the Eli and Edith Broad Center of Regeneration Medicine and Stem Cell Research at the University of California, San Francisco. The center's research efforts are organized in a series of pipelines that each focus on a particular tissue or organ system. These pipelines include both basic and translational research groups working in close collaboration. Dr. Kriegstein's research focuses on the way in which neural stem and progenitor cells in the embryonic brain produce neurons and ways in which this information can be used for cell-based therapies to treat diseases of the neural system. His talk today will touch not just on his own research, but also his personal perspective coming from a developmental biology background into the field of stem cell biology.
Well, thank you. I certainly want to thank the organizers of this wonderful meeting for the opportunity to speak here, and also for Heather for organizing this panel discussion. And as she'd mentioned, although I'm director of a broad stem cell center, I thought I would give you a personal perspective on my view of where stem cell research has gotten us so far, and then perhaps have some opportunity to discuss that if you'd like afterwards. So I thought I'd emphasize what I consider one of the chief achievements of stem cell research, which has been that it's been allowing us to start looking at human cells instead of just animal cells. Because as all of you know, features that are present in rodents are not always shared with people. And that if we're really going to treat human diseases, it's very important to study healthy and also diseased human cells, heart, liver, brain, etc. Something which really hasn't been possible until just now. And this is important, obviously, for a whole range of objectives, including discovering disease biomarkers, creating models of disease mechanisms, discovering new druggable disease targets, and testing therapeutics, of course, both for efficacy and as well as toxicity. And stem cell biology has now allowed us to do this. And I consider this to be one of the major achievements of stem cell research so far. Now, in this audience, I hardly need to discuss how stem cell lines are derived, but I did just want to emphasize how the technology has been moving along so quickly, so that embryonic stem cells traditionally derived from a very early stage blastula embryo produce pluripotent cells that can make all the tissue types in the body. There are adult organs that harbor resident stem cells, so-called adult stem cells. They obviously have more restricted potential. They're multipotential, but not pluripotential. There are adult somatic cells, like skin cells, which can be transformed, as Shinya Yamanaka has shown us, with transcription factor expression into pluripotent cells that are very much like embryonic stem cells. And more recently, it's possible to take a somatic cell, a fully differentiated cell, and directly reprogram it into a cell type of another adult cell tissue. So reprogramming has actually changed the landscape of stem cell research, and by landscape in this case, I'm referring to the Waddington landscape model. As you all know, over 60 years ago, Waddington used a landscape three-dimensional drawing to sort of imply how cells can differentiate. And I wanted to show a film that was actually made by Bruce Conklin at the Gladstone Institute UCSF affiliation that I think illustrates this in a graphic way. Oops, sorry, can we just go back for a second? There we go. And this is a movie. Let's see if we can run this. Yeah, there we go. Terrific. So pluripotent stem cells differentiate as they walk through, or in this case, roll down this landscape to a specific cell type. And in order to induce a regenerated stem cell, you can introduce transcription factors, returning the cells to a pluripotent state. They can then be differentiated down individual lineages. You can directly differentiate these cells down these lineages if you know the appropriate protocols for doing that. And those protocols essentially restrict the fates that these cells can take, for example, in this case, down to heart cells. You can differentiate pluripotent cells into a progenitor state that has a more restricted, more multipotential rather than pluripotential fate. They can be expanded at that stage and then differentiated into specific subtypes of cells like heart and blood vessel stem cells. And then direct reprogramming takes cells that are already differentiated along a particular somatic fate and then transforms them in such a way that they can directly transform from one cell type to another without passing through a pluripotent or multipotential cell state. And these are all tricks that stem cell biology has taught us that we wouldn't have imagined if we just looked at the normal development and the program of developmental organization of the embryo developing toward the fetus and eventually toward differentiated cell types. So I thought I'd use a few examples, starting with cardiomyocytes, because there are protocols available that are really quite robust for generating cardiomyocytes from both mouse and also from humans. And the ability to take human ES or iPS-derived cardiomyocytes creates a really powerful platform for studying a whole range of different disease mechanisms, as well as for refining and improving the ability to develop drugs, as many of you know. So, for example, cardiac arrhythmias, prolonged QT intervals that patients are subject to. Cardiomyocytes have been generated from patients suffering from these inherent genetic diseases. They can be expanded in culture. They can then be derived or differentiated to become human cardiomyocytes. And I'm showing, shamelessly borrowing data here from Christine Mummery, who's taken these ESC-derived cardiomyocytes and cultured them on a microelectrode array chip. And this allows you to actually do field potential recording of the electrical activity of these human cardiomyocytes. And one advantage of this is that you can use high-throughput screening techniques to look at drugs that have toxicity to the heart. And so, for example, two drugs shown here that both have effects on cardiac rhythms have been tested in this in vitro model with the human cells, and the gray bar represents the therapeutic window, that is the blood level or the concentration that has effects in actual patients. And as you can see, the dynamics of the electrophysiology actually starts to alter exactly when those drug concentrations are applied in the dish. And also, when drugs that aren't directly affecting the heart but have side effects on the heart were tested in this in vitro model, as shown in the two graphs down at the bottom, in those therapeutic windows, again, the gray bars, there isn't a change in the rhythm of the cardiomyocytes. But as you increase the dose, you start seeing changes in those electrical rhythms, and those match very nicely the toxicity effects and levels that were found to induce those changes in patients. And so that these techniques of in vitro use of human cells for the first time has the promise of actually allowing us to have better drugs and better pharmaceuticals at a lower price and have more effectiveness when they're finally marketed. But again, I think the greatest contribution of stem cell research is the promise of unlimited numbers of healthy and also diseased human cells for research as well as for therapy. The challenge now is to generate authentic human cells of exactly the right type that you want and also of the appropriate age. So I thought I'd use this opportunity to mention a little bit of the area of interest for my own lab, which is on cortical development. And perhaps nowhere else except in the cerebral cortex can you really be struck at the differences between the rodent models, which most of us use, and the human model that most of us are trying to target. So on the left, of course, to the same scale, is a photograph of the mouse brain on the left and the human brain on the right. And aside from the fact that there's a huge increase in size, I want to emphasize that there's a huge increase in the number of cells found in the cerebral cortex in humans and primates. So on the right-hand side, at the top panel, is a cross-section of the mouse brain, which has a smooth laminar cortex, something called lissencephaly, compared to the human brain, which is shown below, which has a highly folded surface, a gyrencephalic surface, which really allows you to compact a much larger cortical sheet consisting of many, many millions of more cells into the same size or smaller size skull. So this huge expanse in the number of cells in the cerebral cortex underlies not only the abilities that people have for learning and memory and cognition and so on, but also is the seat of a lot of the diseases of cortical function that we're very interested in treating: autism, Alzheimer's, schizophrenia, and so on. So how do these cells arise? And what are the differences between the mouse and the human? Well, the mouse has been studied for the last decade pretty intensively by labs all around the world, and it's becoming clear that there are two kinds of stem cells and progenitor cells found in early cortical development. Radioglial cells, shown here in blue, that produce these intermediate or basal progenitor daughter cells, shown in green, and those daughter cells then make the neurons that are found in most of the nervous system, especially the area of the cerebral cortex that we're talking about. But what about in human brain? Is it the same, or how does it differ? In fact, the human brain has the same two kinds of cells that surround the ventricle that I just mentioned in the mouse. The radial glial cells shown in red, and these daughter cells shown in green, which are these intermediate progenitors. So that looks very much like the mouse. But what the human has that the mouse doesn't have is this huge outer subventricular zone, which is filled with progenitor cells that are proliferating. And these cells are presumably responsible for this huge increase in the cell types that we find in the human brain. So looking more carefully at what these cell types might be, using markers that are specific for the cell types that we've looked at in the mouse, the radial glial stem cells shown in red and the intermediate progenitors shown in green in the mouse are exactly the same as what we find in those same regions in developing human brains. So on the right-hand side, at the ventricular region, you see the red and the green cells representing the neural stem and the neural progenitor cells that are also found in the mouse. But those outer subventricular zone cells, that huge proliferative zone that doesn't exist in the mouse, is also filled with neural stem and neural progenitor cells that hadn't really been described in detail. And so we took advantage of an opportunity to look at these cells, and I wanted to show you a brief time-lapse image of how proliferative this cell region is. So if you could just show this movie, there we go. And the outer subventricular zone in human is filled with these progenitor cells that undergo a very particular kind of division we call mitotic somal translocation that's highly characteristic for just this kind of neural stem cell that hasn't been described in model systems before. And so in the human, there are two kinds of cells that produce most of the neurons in our brain, especially in the cerebral cortex, that aren't found in the mouse and that haven't been modeled in the mouse so far. And those are these ORG cells, the outer radial glial cells, which are neural stem cells, and the IP cells, or the transit amplifying cells, which are the daughters of those cells. And right now we have no markers that can distinguish these human cells from the mouse cells that we've been studying so far. But they clearly are going to be very important if we're going to understand how the brain develops, understand how to make these cells in vitro in a laboratory, or actually be able to use them to understand diseases like schizophrenia, autism, and even Alzheimer's disease. So on the left, I'm showing you the scheme of how neurons are produced in the mouse brain or the rodent brain, where radial glial cells make those more restricted progenitor cells that then make neurons. And in the human, there's an evolutionary advance where that radial glial cell makes a new kind of stem cell, this ORG cell. That ORG cell then proliferates over multiple rounds, makes daughter cells that develop themselves multiple clones of neurons, and greatly increases or amplifies the number of cells that are produced. And this gives us not only the larger brain that we have, but it also highlights the fact that you can't always study, in this case, nervous system development in a mouse model, and that we have to be very careful to use the right models for the diseases or the patients that we're looking for. And when it comes to human disease like this or human development, there really is no substitute for looking at those human cells. On the right, I also wanted to emphasize in this diagram that not only does this outer subventricular zone filled with this new kind of stem cell make the neurons that we find uniquely in the human brain, but they also have these radial fibers, which I haven't emphasized, along which their daughter neurons actually migrate. And so by increasing the number of these cells in the developing brain, you also provide new routes for migration so that more cortical columns can form as the cortex expands. So this understanding of developmental biology is going to have now, I think, an influence on how we make these cells in laboratories to treat or to understand some of these cortically based diseases. And as many of you know, in order to make specific cell types, we've had to use protocols that really are derived from our understanding of normal embryonic development. And when it comes to the nervous system, I'm showing you here on the left the understanding we currently have for how these cortical neurons or forebrain cells derive during normal development. A series of inductive steps, and then there's regionalization. There's a whole set of temporally specific changes that occur. And each of these are signaled by interactions between signaling molecules and receptors. And that understanding of how you go from an undifferentiated cell to a very specific kind of neuron has actually informed the strategies that we've used in the laboratory to make specific cell types, so that we can take the same same growth factors that we know occur to regionalize the forebrain and to create dorsal and ventral patterning, and just replicate that by exposing our undifferentiated stem cells to the same factors step by step, and eventually, if we're lucky, we can actually make the very specific cell type of interest. And I want to give you one example from my own lab and a collaboration I have with some other investigators at UCSF, especially Arturo Alvarez-Bulla, John Rubenstein, and Scott Baraban. And we've been looking at a specific kind of nerve cell known as an inhibitory interneuron that inhibits the activity of excitatory cells. And in normal development, these come from a certain part of the brain called the medial ganglionic eminence, which is actually a factory that just makes these inhibitory interneurons. And from that site, they migrate and disperse to other brain regions. So we hope to use these cells in order to treat diseases, in particular diseases of overactivity or hyperactivity, for example, epilepsy. And in a proof of principle experiment, we've taken these MGE cells, these inhibitory interneurons, from one mouse and grafted them into another mouse's brain, and a mouse, in fact, that has a genetic basis for epilepsy. And the cells shown here in green can migrate, disperse, and integrate, which is a very important feature of nervous system therapeutics, and form connections with the native cells, in this case, throughout the cortex. And in this particular animal model of epilepsy, grafting these cells actually suppresses the seizures clinically and is shown here electrographically as well. But if this kind of an approach is ever going to be used in patients, these cells have to be created out of human cells, not obviously mouse cells. And so we've been working with CERM support on a protocol that actually allows us to take these undifferentiated human pluripotent cells, either embryonic stem cells or iPS cells, and then progressively, step by step, convert them into the interneurons, these MGE-like or inhibitory interneurons that I've shown you that were grafted from the mouse. And we now have large numbers of purified inhibitory interneurons from human cells that we can start using in our animal models. But in doing these studies and looking at studies from other laboratories, there's been a problem that has emerged, and that is that most of the differentiation protocols yield immature or fetal versions of the cells of interest. And this has been reported for ES or IPS-derived cardiomyocytes, hepatocytes, pancreatic islet cells, as well as neurons. And this is an issue that will need to be addressed if you're planning to use the cells either in high-throughput drug screening assays, disease modeling, or, of course, for transplantation therapy. For example, in the cells that I've just mentioned so far, we've been looking at how long it takes for these cells to mature. And in fact, to go from the migratory neuron stage, which is shown here in the photograph on the left in blue, to a differentiated cell that has all the electrophysiological properties of these mature interneurons can take 6 or 7 months for these human cells, which seemed like a very long time. So we went back and looked at fetal human development and asked how long does it take for these cells to develop normally, and it takes 6 or 7 months from the time they originally are derived or born till when they actually start functioning just like these in vitro cells. So we aren't altering the genetic or the developmental pattern at all. It's just unraveling in the human time scale. And in contrast, these same cells that we've derived from a mouse will mature within a month or so, 4 to 5 weeks. So the mice obviously has a very much faster timetable for differentiation than humans do, and that's an issue that I think many of us are going to have to address. The other issue about human and animal models I wanted to mention is that when you're basing clinical trials on disease-modifying activity in a non-human species, for example in the mouse, there are significant differences that need to be addressed, especially when you're talking about the nervous system. The brain, the cortex, or the spinal cord in humans is very different than the brain or the spinal cord in a mouse or a quadruped. And I just wanted to cite one historical example where going from an animal model, especially a rodent model, into human trials has not worked out very well. And that's when, as many of you may know, the experience with the models of NMDA-induced excitotoxicity, which were very widely accepted as the mechanism for damage and stroke and traumatic brain injury, led to a whole range of therapeutic approaches that worked very nicely in animal models. And hundreds of millions of dollars were actually spent in taking these animal models and translating them to the clinic, and essentially none of them worked. I've just put a list of some of the drugs that failed here: lubeluzole, elaprotal, sulfatil, Degeneration, etc. Most of us don't remember these drugs. And the amount of time, effort, money, and resources that were spent on pursuing this, was really, I think, a lesson to all of us that we need better models if we're really going to try to address this problem in a way that is more efficient and more cost-effective. So my final remarks then are along those lines. I don't want to leave you with the impression that we have to use human cells only and that the time has come to abandon animal work and turn entirely to the use of these human cells that we can now produce in our laboratories. And I wanted to just emphasize that nearly every major stem cell breakthrough has been made in the mouse first and then later on in the human, and that's not by accident. Mouse models are cheaper. They're much faster. I've given you an example of how much more quickly cells mature in the mouse than, for example, in the human. And they provide the proof of principle experiments that are really critical before we move into large animals or certainly into people. But the emergence of stem cell-derived specific human cell types in vast quantities, which we can now make, provides us really an unprecedented opportunity to tackle human development and disease in a way that just couldn't have happened before. And this promises, of course, to accelerate clinical breakthroughs, provide safer, cheaper, and more effective therapeutics. And in my view, this is really what the revolution of stem cell biology is really all about. And I think there was just one more slide. There it is. Yes, I'm very proud of this CERM-sponsored building, which is the home of our program at UCSF. And any of you who are visiting San Francisco in the future, I'd certainly invite you to visit. I'm happy to show it off. Thanks so much for your attention.
Thank you. And for our third speaker, I'd like to introduce Dr. Lawrence Goldstein. He's the Professor of Cellular and Molecular Medicine at the University of California, San Diego School of Medicine, and he's the Director of the San Diego Stem Cell Program. His research is focused on understanding the molecular mechanisms of movement inside brain cells and how failure in these movement systems may lead to neurodegenerative diseases. Dr. Goldstein has had an active role in national science policy and was also co-chair of the Scientific Advisory Committee to the Proposition 71 campaign. He is a strong advocate for the responsible introduction of new stem cell treatments into the clinic, and here he is.
Good morning. So the bad news is we're way behind schedule. The good news is I'm going to try to get us back on schedule. So I think it's really important for me to try to leave us with some time for questions and discussion from the audience. So I'm going to give you a somewhat abbreviated version of what I was going to tell you about. A little bit less detail, but nonetheless emphasizing the concepts that I think are crucial for an audience such as this one. Now, what the theme will be of my remarks here and then subsequently after the break in a talk I'll give, is the notion of mechanism-based, evidence-based development of medical therapies. Because I think that's really crucial. It's very easy to forget how important it is when we move things into humans to try them out on people, that we have a very sound foundation moving forward so that A, we don't hurt anybody any more than is necessary, and B, we have a reasonable expectation of success as we move forward. Now, the analogy I love to start with is one that goes back to my years as a teenager when I owned my first used automobile. And when it would break, I would try to fix it. And at first, that was a pretty iffy proposition, usually resulting in a trip to the auto mechanics, because I didn't know the first thing about how an internal combustion engine worked and what all those parts were when you opened the hood and looked inside. I'm still a little shaky on it, but I'm doing better. Now, after a few years of working on internal combustion engines, I actually learned something about the principles by which they worked so that I could begin to recognize the sounds and symptoms of different kinds of abnormalities in the engine. For example, when you have an engine that's missing on one cylinder, you know to start looking for things, or when spark plugs get fouled or what have you. I guess cars don't have spark plugs anymore, though, do they? I've now dated myself. But the point is, I did a much better job of repairing the automobile engine after I understood something about how it worked. Now, this morning, as an example of how that translates to medical therapies, the Nobel Prize in Medicine was awarded to an old friend of mine, Bruce Beutler, who is not only a UC San Diego alumnus, as Ann Tsukamoto and I both are, plug for my university there, but Bruce's work in the 90s on trying to understand how inflammation worked led directly to the development of a drug called Enbrel or etanercept, which is one of the most effective drugs for the treatment of rheumatoid arthritis, psoriatic arthritis, and the spondyloarthropathies as a whole. And the point is, that drug would not have been developed without Bruce's curiosity and drive to understand how inflammation works in mice and ultimately humans. And so I want to take that idea and now apply it to Lou Gehrig's disease, which is a disorder where we are hopeful that stem cell therapies will help us make an impact in the coming years. So let's begin with a reminder. Okay, let's get organized up here. How's this? Okay. With a reminder of the disorder, when Lou Gehrig developed it, he had a pretty typical course of disease. It started with painless muscle weakness, which he primarily noticed because it affected his golf game and slowed him down when he was running out of hit. So he got to first base a little bit more slowly. Let's see, how do I use this thing? Okay, I have too many devices up here. All right, next. Okay, I can get that. So the disorder is a simple one to describe because it's a disorder where one kind of cell inexorably dies, and that's the motor neuron, which is the cell that's responsible for sending the voluntary movement signals from the brain and spinal cord out to the periphery. So when I move my fingers, I'm using my motor neurons that run from my spinal cord into the muscles in my fingers to generate those movements. And what happens in Lou Gehrig's disease is those neurons die for reasons we don't completely understand. And the really rotten news is there is really no effective therapy for this disease, so that a typical course from diagnosis of the disease till death is about 3 to 5 years, with some forms of the disease leading to death within 1 year of diagnosis. It can be very aggressive. There's a long list of drugs that have been tried in clinical trials, and only one of them has achieved approval, this drug Riluzole, and that drug barely makes a difference at all. If you study enough patients, you can see that a small number of them have a few more months of life with this drug, statistically speaking, but individual patients don't notice a difference. And so the point is, we must do better to find a way to treat this terrible disorder. Now, the point I want to make is ultimately gaining a deep understanding of what happens in Lou Gehrig's disease is likely to put us on the path to an effective therapy. And that's what I want to illustrate for you in the next 5 minutes. And the point is that although most Lou Gehrig's disease is what we call sporadic, meaning that we don't entirely understand what causes the disorder, there are rare forms that are hereditary in a simple way, and one of those forms is caused by a genetic change in a gene that makes a factor called superoxide dismutase, or SOD1. And so all you need to remember is that damaged SOD1 will generate aggressive Lou Gehrig's disease in humans, and when you put that damaged SOD1 into mice, you get a disease in mice which looks very much like human Lou Gehrig's disease, well, except, of course, mice don't play golf. Now, what that kind of mouse model lets you do is to ask an important question. So most of us, if we were to be asked the question, if we wanted to develop a stem cell therapy for Lou Gehrig's disease, what kind of cell should we make to transplant into these patients? And your knee-jerk reaction would be, the motor neurons are dying, let's make new motor neurons and replace them. But the problem is that, A, those cells, as I just mentioned to you, are very long. They wire all the way from the spinal cord to the fingers, the diaphragm, the legs. Really, really tough wiring problem. And second, what if the motor neurons aren't dying because they themselves are defective? What if another cell is defective and is killing them? Now, you might think that that's a theoretical issue, but in fact, in type 1 diabetes, so-called juvenile diabetes, what happens is that beta cells in the pancreas die, but not because there's something wrong with the beta cells. It's because the immune system becomes abnormal and attacks them and kills them. So, how can you solve that kind of issue in Lou Gehrig's disease using these mice? So imagine for a moment that you could do really sophisticated genetic experiments in the mouse, which you can, so that you could make mice that are a mixture of Lou Gehrig's disease mutant cells and normal cells. And so we'll have red equal the Lou Gehrig's mutant cells and the green equal normal. And so you could ask the question, what if you had one motor neuron in red here that had the Lou Gehrig's disease mutant surrounded by normal cells? Would it survive better than normal? Or, alternatively, what would happen if you had a normal motor neuron surrounded by Lou Gehrig's disease mutant cells? Because remember, the spinal cord is not just composed of motor neurons. There are astrocytes, there are glia, there's all sorts of other cells there. Maybe somebody else is doing something bad. So, 10 years of work later, from my lab and my colleagues' lab, Don Cleveland, that's what all that stuff is, we ultimately got to the point where, let's see, which would I like to do? Oh, let's do this. Yeah, okay, there we go. Where what we learned is that although the motor neuron itself contributes to the disease, the surrounding cells, in particular a cell type called an astrocyte, can provide a substantial part of the so-called killing activity from Lou Gehrig's mutants, as well as rescue activity if they're normal. And that's true in mouse models of Lou Gehrig's disease tested a bunch of different ways. It's true in transplantation experiments done by my colleague Martin Marsala in collaboration with a company called NeuralStem, using transplants of fetal spinal stem cells into rat models of Lou Gehrig's disease. And in fact, it's true in multiple experiments with purified cells done outside of animals in vitro, that is, in a lab cultured dish. And as a result, we're in the midst of a 4-year project, we in this case being myself, Don Cleveland, Life Technologies, Sam Pfaff at the Salk, and Martin Marsala at UC San Diego, to use embryonic stem cells to develop astrocyte progenitors for testing and ultimately for clinical trials using transplantation into human patients. And the idea is really simple. Start with human embryonic stem cells, expand and grow, induce differentiation to astrocyte progenitors. I won't take you through all of the gory details, and then develop a clinical trial based on transplantation into either cervical or lumbar spinal cord. Now, Ann Tsukamoto gave you, I think, a pretty good understanding of what the challenges are in developing a production process, and so what we're working on right now is developing a rigorous, safe manufacturing process where not only do we start with well-characterized embryonic stem cells, but at each step through the process of generating the cells that we ultimately want to transplant into humans, we have verifiable and rigorous information about what cell type we've grown in the dish, and that we can make it reproducibly and reliably so that every time we test it, we get a consistent kind of outcome. And that's really important, and it's something that's a very important part of working in well-regulated environments, such as the US and Europe and Japan. That is, we have accountability that the cells we make, or this is true in the drug industry, are exactly what we say they are, and we can prove it. Okay, so let me just mention that at present, we're working on developing an embryonic stem cell-based approach to this disorder. There are currently clinical trials also using umbilical cord or autologous blood stem cells at a couple of sites, and the neural stem cells that I mentioned are being tested in a trial at Emory University. And I just want to point out that if you go to the literature and ask the question, is there evidence as strong as what we and colleagues have produced that astrocyte progenitors are likely to be a beneficial cell type. That is there comparably strong evidence for blood stem cells or mesenchymal stem cells or what have you for Lou Gehrig's disease, although there is the occasional article that reports some benefit and certainly safety, the evidence is simply not as strong. And I just point that out because there is a tendency, if there is a paper or two in the scientific literature that reports some benefit with one kind of stem cell or another, for folks to say, see, it's working, it must be that it's now safe for use in humans. And that's not true. There is a real benefit to having rigorous, reliable, statistically significant evidence from a broad range of studies. Now, I want to point one other thing out, and that has to do with this study using blood stem cells at this company, TCA in Louisiana. And that is, this company, which has been running these phase 1 safety trials, recently got a very stern warning letter from the FDA because they had violated a number of the methods and approaches in their protocols. For example, some patients who were supposed to get stem cells from themselves by mistake got stem cells from other donors, or things weren't tested for viruses as carefully as they needed to be. Now, my point here is not to take TCA to task, because TCA is going to clean up its act having gotten this letter from the FDA, I'm sure. The point is, one of the benefits, again, of trials in a well-regulated environment is that when there are problems like this, they get caught and they get fixed. If this had been a therapy being delivered in a poorly regulated environment, you would never know if the cells you were getting were exactly what they were supposed to be, whether there'd been a mistake, what have you. I mean, you would never tolerate that kind of behavior out of an automobile shop down the street from you. If they were transplanting motorcycle parts into your Cadillac, for example, that would be a bad thing, and you want accountability. And my point is that a well-regulated environment helps to generate that kind of accountability. Okay, final two issues, and then we'll move to the panel. One issue is people sometimes say, oh, well, we know that fat stem cells or blood stem cells or this kind of stem cell or that kind of stem cell is, quote, safe because we've been using it for a number of years. And that's true in a sense. But remember, when you put cells from your body into new environments, they don't necessarily behave the same way as they do in their natural environment. And of course, the thing we always worry about, and we want to be sure we have evidence about before we proceed, is what if the cells don't stay where you put them? What if they do something bad after you've introduced them? And evidence is starting to accumulate from some of these trials or companies that are marketing therapies that are not well supported by careful, rigorous scientific evidence that, in fact, bad things can happen. Tumors are starting to appear, destruction of tissue, and a couple of deaths as well. And I guarantee you, if I were to want to try to treat Roman Reed with cells I had generated in my lab, I know how anxious Roman is to get out of that chair, believe me. But I also know that he and his dad, Don, don't want me to do something to Roman that's going to make him worse. And that's the thing that we have to be really cautious about in our drive and desire, legitimately, to move things as rapidly as possible. We have to not make people worse. Okay, final issue. The problem of clinics selling therapies in poorly regulated markets is almost overwhelming at this point. If you type the name of your disease and stem cells into Google, you will come up with tons of clinics offshore who for $50,000, $60,000, $100,000 will sell you a cure. Now, how do you know it's a cure? They say it is. But what about being a little skeptical? And the problem we sometimes have is that if one person goes to one of these clinics, or let me put it a different way. Let's suppose 1,000 people go to one of these clinics, and one person gets better, and that person's testimonial appears on the clinic's website. Does that constitute evidence that this is the place that's going to deliver a therapy that's really going to work? It's a problem of testimonials. What happened to the other 999 people? You don't know. And the issue that we now face is sometimes just because a therapy is in a phase 1 clinical trial for safety, companies start using that as an excuse to market their therapy as a proven treatment. And of course, this was recently highlighted in a 60 Minutes program last year called 21st Century Snake Oil. I was privileged to be a part of this. It won an Emmy recently for CBS. And the point is, there really are charlatans out there taking advantage of people's desperation and lack of information about how medical therapies are developed and how they work. Not everybody is a snake oil purveyor, but in fact, when Peyton Manning recently went to get stem cell therapy, or Rick Perry, their prominence in the community really makes this problem worse. Because if Peyton Manning had happened to get better, I'd argue we don't know whether the stem cell treatment is what made him better or not, because he has the best medical care in the world. He is the most disciplined athlete on the planet. You have to have large-scale clinical trials to really test whether these things work or not, and whether they're superior to just normal physical therapy. That's ultimately the issue. So my final point, and then I'll turn us over to the panel. I find that I'm offended by something that has developed over the past several years. And what has developed is that opponents of embryonic stem cell research, that is, those who have trouble with the ethics of embryonic stem cell research, often point to these snake oil treatments as evidence that embryonic stem cells are not needed to make medical progress. Now, let me just say, I respect people who think that embryonic stem cell research is unethical. I disagree with that point of view, but I understand why they might think it's unethical. But let me just say that I think it is more unethical to tell people, to encourage people to go to these offshore snake oil clinics and pay large sums of money for bogus treatments or treatments that might make their family members who have a disease worse. Thank you.
Thank you, Larry. So I'm just going to invite the panel to come back out, and we're running a little bit over time due to a late start, so we're probably only going to get 1 or 2 questions in, but if you've got a question, raise your hand, and there are a couple of roaming mics. So one. Okay, do we have the mics on? I'll assume so. So I think I'll start off, and if anyone else has a question which we can see, I'll try and look out. I don't have the greatest visibility. So we've heard from each of the panelists today, and one of the things that we've heard about is the importance of the strong evidence going into a clinical trial and going into the clinic. But this takes a lot of time, and we also heard from CERN saying that they're wanting to get out there and talk to the public and make sure that a strong message is going forward. So when we're talking to the public, what can we say?
What should we say, or what can we say? Okay, so look. I think the bottom line, in my own personal opinion, I don't know if anybody will agree with me, is we have a responsibility as scientists to move our understanding and discoveries to clinical application as rapidly as possible. Every one of us, whether we're scientists or not, have family members who are sick, who have diseases where we want to deliver a therapy as rapidly as possible. But that doesn't mean we can make progress by cutting corners. We need to be, let me put it a different way. I sometimes use the term aggressive restraint. So the aggressive part is we want to just break all speed records to make it to clinical application. But we have to rely on the judgment not just of ourselves, but of our regulatory system and our colleagues to ensure that our absolutely furious race to the clinic also doesn't make things worse. And while it's very easy, for example, to test a piece of software or an Intel chip in a very short amount of time in a laboratory setting, human experiments and trials take time. If you want to know if a drug or a treatment for Alzheimer's disease is working in a clinical trial, it means following people for several years to be sure they don't get sicker from what you did and that they really improve. Yeah.
I believe there's a question from the audience.
Dr. Goldstein, as a scientist, I completely understand what you're saying, but as a patient with Parkinson's disease, I don't have the luxury of waiting for 10 years. What am I, how do I resolve this conflict? And I keep telling, I'm a patient advocate. I tell my fellow Parkinson patients, please don't go for all these therapies, but they have no other hope. And if it works 1 in 10, for whatever reasons, doesn't matter what the mechanism is, people are willing to take the chance. I don't know how to answer those people. They say 60,000 is going to get me a remote chance of getting up and walking down the aisle with my daughter. I would do that, is what one person told me. As a patient, I completely understand the sentiment. And as a scientist, just like you're saying, I was working on AIDS, and I was telling AIDS patients, please wait. And I didn't realize the constraints of time. Now, being on the other side, I totally understand how patients feel. And I mean, this is a question for anybody who is working on not only stem cells for anything else, but...
So I completely agree with you. It is an extremely difficult problem. And I also have family members who have been sick over the years, and I've gotten to know a great many patients with Parkinson's disease and ALS. But I think we have to realize that there are two sides to the, quote, therapies that are for sale around the world. Or actually multiple sides. First, there's generally no evidence that they'll actually work. And second, there's actually evidence developing that they will make people worse. And so I think the question is, where does any individual patient draw the line on how much evidence they want to have before they decide to undergo either an experimental therapy in a clinical trial or to go to a poorly regulated environment and pay a large amount of money for a, quote, treatment where there's no evidence that it will work? I mean, I think that is an individual decision, and what I think I would urge all of us to do is to pressure all of these clinics to really provide transparent, verifiable information about what they're doing to patients. And then if people feel that the evidence is strong enough for them to pay for a therapy, that's an individual choice. But I think that from a regulatory standpoint, we want to really push for transparency and verification that people are telling you the truth.
Perhaps you could, sorry.
Well, I was just going to certainly endorse all of those comments, and I feel that it's important that the public should understand that only by doing clinical controlled trials will we really get to the bottom of whether any of these therapies work or how well they work. Anecdotal remarks are just not adequate. Even the phase 1 trials that are ongoing now, for instance, in spinal cord injury and so on, might result in a patient or so saying that they've noticed improvement. Anything along those lines is always going to have to wait until there's a proper controlled trial before we really know if these approaches are actually going to work or not. And CIRM has been terrific in actually pushing the agenda of translation as hard as they have. I think perhaps one of their biggest legacies so far has been the creation of these teams and the notion that in California, so many even of my colleagues at UCSF are thinking about translation in ways they hadn't before. And I understand the patients are very impatient. I'm a neurologist, and I've seen so many patients who've passed away with some of these neurodegenerative disorders that have really no hope. But we just have to be patient. These diseases often have placebo effects that are just, especially in Parkinson's disease, that are just unbelievable. Patients who actually have PET scans that show increased dopamine when they were told that they were given a therapy and it turned out to be a placebo. So unless you look at properly controlled trials, we're just never going to know. And we aren't quite there yet, and I think we all have to be patient.
So, Anne, I'll give you the last word before we pass back to Bernie.
I agree with all that was said by both Larry and Arnold. And I think I want to add that one of the reasons we are doing our clinical trial in Switzerland is because all of the points that were made and all of the items that we need to move forward as quickly as we can lined up in Switzerland. We're in this trial, as I mentioned, able to do patients in cohorts who have an Asia score of A, B, and C, as opposed to doing that, starting with A, completing that trial, moving to Bs, and starting another trial, moving to Cs. The timeline to complete those trials would be enormous. If you can complete your phase 1 safety studies in these patient populations in a quicker time and show that it's safe, and then move into a trial like a phase 2 or a control trial where you really know that what you're seeing is the effect of the cells and not some placebo effect, I think we will get to those points faster. And I do agree with the comments that were made about all the unproven therapies are taking the advocates and the patients and the funding that could be going for the solid, reputable work, taking them and putting those efforts into things that have less chance of working, that could cause harm.
Thank you. And thank you all for joining us today. Bernie?